أعوذ بالله من الشيطان اللعين الرجيم بسم الله الرحمن الرحيم اللهم صل على محمد وآل محمد وعجل فرجهم. Friends, today we are going to discuss reverse transcriptase polymerase chain reaction. Sometimes some people confuse this reverse transcriptase polymerase chain reaction with real-time polymerase chain reaction. Actually, real-time polymerase chain reaction is quantitative polymerase chain reaction, but now these two techniques can be combined with each other, and real-time reverse transcriptase polymerase chain reaction can be performed. Actually, when our available product is RNA and we have to amplify this, then this reverse transcriptase polymerase chain reaction is performed. But conventionally, for PCR polymerase chain reactions, DNA is the product. DNA is the product which has to be amplified, which has to be extended. But here, for in reverse transcriptase polymerase chain reaction, we have RNA which has to be amplified. There are two main types of reverse transcriptase polymerase chain reaction. Number one step RT-PCR, and number two-step RT-PCR. One-step RT-PCR is performed in single tube, and in single tube all the reactions takes place. All the reactions means, first of all, RNA is converted into complementary DNA. Complementary DNA is formed at first step, and this complementary DNA act as template, and this formation of complementary DNA or cDNA is taken place by the help of reverse transcriptase enzyme. When complementary DNA is achieved, and this whole process of reverse transcription is taken place in one cycle in thermocycler at 50 degree centigrade temperature. And when this complementary DNA is formed, this step also involves three main steps. Number one, reverse transcription occurs. Number two, annealing occur. Annealing means the attachment of the primer with the messenger RNA to form complementary DNA. And then attach after annealing step, next step is the extension or amplification process. So when these three steps complete in one cycle in thermocycler, then the whole process of reverse transcription completes. After that, we add, if we are performing this whole step in a single tube, in one step PCR, then all the things have to be added in single tube, single polymerase chain reaction tube. And all those things include, as I said, that number one, the primers for the formation of complementary DNA. Complementary DNA is formed from messenger RNA or total RNA. Number two thing which has to be added should be Taq polymerase enzyme when we achieve complementary DNA. Next, this complementary DNA is further has passed through 30 to 40 cycles in the thermocycler, and again three main steps undergone there. And those three steps which undergone there, they are number one step is the heating or denaturation process. Number two, is the annealing process, and number 3 is the extension process. So complementary DNA also undergoes all these three steps, and then complete product is obtained. So this is one step process. In two-step process, reverse transcription process is completed in a separate tube, and the next PCR process is completed in another tube. So we can explain it like this, that first of all, messenger RNA or total RNA is converted into complementary DNA by the help of reverse transcriptase enzyme in a separate tube. And when we achieve complementary DNA, this complementary DNA is again added in another tube, and in that tube we add primers. For these specific primers, gene specific primers are added in this process in a second tube. So again, denaturation of the DNA occurs at 94 to 96 degree centigrade temperature. Number 2, annealing process occurs at 50 to 56 degree centigrade temperature. And last one is the extension process, which occurs at 72 degree centigrade temperature. So these are the main steps which are involved in RT-PCR process. RT-PCR can be used for the identification of RNA from the virus, mainly viral disease detections. Now, scientists are trying to detect cancer as well from this RT-PCR, and so many other diseases like genetic diseases, they are also being identified through RT-PCR. Jazakallahu khaira. Allahumma salli ala Muhammadin wa ali Muhammad wa ajil farajahum.